Well, hello, my fellow pre-meds and pre-health students more generally. Welcome back and congrats on making it to the second part of our carbohydrate structure and function video. So in the previous video, we talked about monosaccharides. In this video, we're specifically going to be focusing on disaccharides and polysaccharides. This video will hopefully be easier to digest compared to monosaccharides because with the monosaccharides, we had to discuss a lot of stereochemistry and organic chemistry review as it pertains to monosaccharides. And in this video, to the extent that we do talk about stereochemistry, it's just really going to be building up on those foundations that we've already built in the first video. These videos, at least as of now, are sponsored by you. So if you're interested in supporting me to be able to continue to make these high quality MCAT playlists, you can support me either via Patreon or by buying me a coffee. Links in the description down below, as well as the links for everything else, including the link to our growing pre-med companion Discord community. All right, so it looks like we're starting off right off the bat with this very messy slide, and I know I said it's going to be easier, so bear with me. I'm going to break this down to you in a very simple to understand way. So the formation of the glycosidic bond is the basis of forming disaccharides and polysaccharides. And that's because disaccharides and polysaccharides are just monosaccharides attached to one another via glycosidic bonds. Now, glycosidic bonds don't only occur between two sugar molecules, they occur whenever an anomeric carbon, and recall from our monosaccharides video, the anomeric carbon is that first carbon after the oxygen atom in your cyclic sugar structure over there. So when an anomeric carbon encounters a hydroxyl group on any other organic molecule, there's a potential of formation of a glycosidic bond. So if you have a methanol in that case, which is an organic molecule, or ethanol or propanol, any alcohol, you'll be able to form a glycoside. So the outcome of this reaction will be called a glycoside. And by the way, this is a dehydration or a condensation reaction because you have the release of water as part of the product. Now notice the nomenclature over here, you're essentially adding that side suffix, and then the prefix is going to be the name of the alcohol. Now what's more important for the purposes of the MCAT is when you form a glycosidic bond between two sugars, because sugars are organic molecules and they contain OH groups. So they can also be on the receiving end of that glycosidic bond formation. So you can have alpha-D-glucose, which is contributing its anomeric carbon, and then beta-D-glucose over here, or even alpha-D-glucose or any other cyclic sugar molecule that can contribute one of its hydroxyl groups to form a disaccharide. But of course, a disaccharide contains a glycosidic bond within it. And that glycosidic bond you can see right here is a carbon-oxygen-carbon bond. Now, in this case specifically, when you combine two D-glucose molecules, you get maltose. And maltose is connected via an alpha-1,4 glycosidic linkage. More on the naming in a bit, but just so you know, so like this is alpha-1, the first carbon, and then it's attaching to the fourth carbon of the second glucose molecule. So 1, 2, 3, 4. Okay, and so polysaccharide formation is essentially the same process. You're just forming more and more glycosidic bonds. Now, the way you break a glycosidic bond is via hydrolysis, because the idea over here was that when you formed it, you release water. So if you want to break it, you add water. All right, so now more on disaccharides. So again, disaccharides are formed with the interaction of two cyclic sugar molecules. And what's important when it comes to glycosidic linkages or glycosidic bonds is that you first identify, and this is the naming system, very, very important, okay, on the MCAT. So you first want to identify whether you have an alpha or beta anomeric carbon. And then you want to identify the number of your anomeric carbon, which is typically going to be 1, and then the number of your hydroxyl group, which can be 4, 6, sometimes carbon number 2. So that's the carbon on the second cyclic structure over there. Now, and we'll go over these examples in a bit, but one exception is when you have two anomeric carbons interacting with one another. In that case, you have to name it based on both. So you have to mention, for example, instead of just mentioning one anomeric carbon, for example, alpha-1,4, here you would say beta-beta-1,1, or alpha-beta-1,1, or alpha-alpha-1,1. So that's when two anomeric carbons join one another. Although the common examples on the MCAT only have one anomeric carbon involved, as we'll see right here. So this is the structure of sucrose, and sucrose is formed between a glucose molecule and a fructose molecule. And it's specifically formed between the alpha-1 anomeric carbon over here, so you can see glucose alpha-1, and the number 2 hydroxyl group in fructose. So it's glucose alpha 1, 2 fructose. Okay? If you look at maltose, it's formed between two glucose molecules. So you have glucose alpha 1, 4. This alpha 1 part over here, and then number 4, if you count 1, 2, 3, 4, then you have alpha 1, 4 glucose. Okay, and then for lactose, again, another common one, you have galactose, beta 1, 4 glucose. So you'll notice that this is the anomeric carbon, and it's in the beta position. So that's why we say beta 1. And then again, 1, 2, 3, 4 is the fourth carbon atom on glucose. Now, one thing that might be a bit tricky is that over here, we start counting to the right of the oxygen atom, whereas here you're counting towards the left. And to be honest, it's a bit confusing. You can draw out the structure of fructose and see why, like the Fisher projection, and see why this is the case. But honestly, whenever you see glucose and fructose attached to one another, automatically on the MCAT, you should recognize that as sucrose, and you should know that it's connected by an alpha 1, 2 glycosidic bond linkage. And then one note on lactose, which highlights the specificity of enzyme functions. So recall from our enzyme video, we talked about how specific enzymes are for their substrates. So some of us will lack the enzyme that metabolizes lactose, which is the enzyme lactase, or we might not have enough of it. So you start to have some GI symptoms. Now, notably, those folks can still metabolize sucrose and maltose, and that's because there's different enzymes involved in the metabolism of these molecules. And although lactose is very similar to the other sugars, the enzymes are highly specific to the point where you can't metabolize the beta 1, 4 glycosidic bond, but you can, for example, metabolize the alpha 1, 4 glycosidic bond. Okay, so now let's talk about polysaccharides. So polysaccharides are essentially long chains of monosaccharides. So disaccharides are 2, polysaccharides are long chain. Oligosaccharides you'll sometimes see are like shorter polysaccharides, like closer to 4 to 5 to 6 monosaccharides connected by glycosidic bonds. And then you have homopolysaccharides and heteropolysaccharides. Homopolysaccharides just means that you have only one unique sugar comprising the entire chain. So like, let's say just glucose or just galactose. Whereas in the case of heteropolysaccharides, you'll have different kinds. So you might encounter glucose, 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 and then galactose, and then mannose, and then fructose, and then glucose, glucose, and then fructose, fructose, and so on. Now, one thing about polysaccharides that's important to note is that they can either be linear, meaning they're like just a straight chain, or they can have branches coming off of that chain. And more on that in the subsequent slides. But essentially, you're forming a glycosidic bond between the kind of the straight chain you have, and then the branches are going to be other glycoside bonds that will allow you to form those branches. Now, the major biological polysaccharides include cellulose, starch, and glycogen, and these are the ones you're most likely to encounter on the MCAT. So let's go over them one by one. Let's first talk about cellulose. So cellulose is a structural carbohydrate, so it helps in the formation of the plant cell wall. And the way it's formed is by linkages. So it's a homopolysaccharide, so only one type of molecule in it, which is beta-D-glucose. And they're connected via beta-1 glycosidic linkages, as you can see right here. So this is, beta-1,4 glycosidic linkages. So you can see right here, this is your position number 1 carbon because it's adjacent to that oxygen atom. And then it's attached to number 4 on the subsequent glucose molecule. So you can count 1, 2, 3, 4, and so on. This is number 1 attached to number 4 on the subsequent beta-D-glucose molecule. Now, this, again, because it's a structural polysaccharide, demonstrates high levels of hydrogen bonding to provide strength and rigidity. Notably, humans cannot digest this sort of polysaccharide, and that's because of the beta 1,4 glycosidic bonds. So as we'll see later, when it's an alpha 1,4 glycosidic bond, we are capable of digesting it. So humans cannot digest this. So it actually serves as a dietary fiber. So you won't be able to derive nutrients from it, but it'll help you with digestion because of the dietary fibers in it. Okay, now let's move on to starches. And notice that starches, first of all, they're the primary storage form of carbohydrate in plants, and then they are also composed of D-glucose, but now instead of beta-D-glucose, it's alpha D-glucose. So that makes a world of difference in terms of how we can digest it. And this again just goes to show the level of specificity of enzymes. Like we're able to metabolize or break down an alpha 1,4 glycosidic bond, but we're not able to break down a beta 1,4 glycosidic bond. What do we mean by alpha or beta? I can give you a brief explanation, but if you want a detailed explanation, you'll probably have to watch our video on monosaccharides if you haven't yet. But essentially, if you're looking at this anomeric carbon right here, and specifically in the chair conformation, if the OH, so over here you can imagine if this was all separated, you'd have an OH over here. If it's going in the axial position, so like vertically down, then you have the alpha molecule, and if it's going to the side, then you have, in the equatorial position, then you have a beta molecule. So in either case, this is formed of alpha 1 glycosidic bonds. So it's a straight chain in the case of amylose. You don't have any branches or a linear polymer. And in the case of amylopectin, it still forms those alpha 1 linkages. However, you have branches coming off at the alpha 1,6 position. So you can see right here, this is your straight chain. So if you number them, 1, 2, 3, 4, 5, 6. So these are the carbons of this glucose molecule. You can see that it's attached to its neighbor via a 1,4 glycosidic bond, alpha 1,4, because this is 1, 2, 3, 4, so fourth position. And this one is attached at the 1,4 with its neighbor, whereas the one that's branching off over here is attached via an alpha 1,6 glycosidic bond. So that's just the way to form the branches over there. And then finally, iodine can detect starches. So if you had a test tube, let's say, and you wanted to determine whether it contains starches or not, if you add iodine, it'll change color, and then you'll know that it tested positive for starches. You may have tried this experiment in elementary school or middle school, where you get potatoes, which are rich in starches, and put iodine on them, and then they'll change color and become like a purplish color. Okay, now let's talk about glycogen, which is the primary storage polysaccharide in animals. So very, very similar to amylopectin. Recall amylopectin, which is a form of starches, we mentioned has those alpha 1,4 glycosidic bonds between alpha D-glucose molecules, and it also consists of the alpha 1,6 glycosidic branching bonds. However, in this case, you have more of those branches. So there's a lot more branching when it comes to glycogen, and that's because the branching allows you, the glycogen to be more soluble and easier to mobilize, so you can mobilize it far more rapidly. And this is quite important because presumably animals will need a more rapid release of glucose compared to plants, such as in the case of fight or flight. You know, plants aren't getting up and running away from a lion, but animals are. Okay, so glycogen is primarily stored in the liver and skeletal muscles. Now let's take a look at the structure of glycogen over here. So you can see this is the extensive branching that you see in glycogen. So you see, like, let's say this is your chain right here, and then you see branches coming off here and here and here. And so you have a bunch of branches. And then you can just look at if you want to zoom in on this part over here, you can see, again, very similar to what we saw with amylopectin. They're attached via alpha 1,4 glycosidic bonds in the linear chain, and then the branches coming off are going to be alpha 1,6 glycosidic bonds. And the idea why, if you want to wrap your head around this, the idea why you can mobilize these glucose molecules more quickly is because if you had one linear chain, you would probably only have one enzyme, or this is the case, you'd have one enzyme detaching one glucose molecule, and then the next, and then the next. Whereas when you have branching, you can have one enzyme here, one here, one here, one here, one here. And so you have a lot more glucose molecules being mobilized. Okay, so this slide is just a comparison between cellulose, amylose, amylopectin, and glycogen. You can pause the video, screenshot it, or later on access the slides and make Anki cards using it, although we do have Anki cards associated with the slide deck, so definitely check them out. But I'm just gonna move past it. Let's do some knowledge checks. And again, I'm just gonna show you the questions and the answers. If you feel like you need any explanations, you can always reach out to me and I'll explain the answers to you. So again, you can pause the video. I'm just gonna speed through these. There's your answer over there. Again, 3, 2, 1. There's your answer. And our last question, 3, 2, 1. All right, so what did you think? Did you feel like this video was easier to digest compared to the monosaccharides? video. I always felt like disaccharides and polysaccharides are a lot more intuitive than monosaccharides. But if you do have any questions, be sure to reach out. You got this. Go crush that MCAT. Did you know that every single video has customized Anki cards associated with it that cover the concepts that we discuss in a video? So if you want a way to be able to actively review the material and retain it for the long run, I've got you covered. If you're interested in accessing the Anki cards or even the slide decks within the videos, then check out our Patreon or reach out to me directly for alternative ways to access the Anki cards and the slide deck. Links in the description down below, as well as the links for everything else.